Now moving to day 2, and we have 5 lectures that we'll be going through. The first one is on novel technologies and methods for NMR-based metabolomics. So yesterday we talked about some of the more conventional methods that are done, the general protocol and workflow for NMR-based metabolomics. And this time we're going to look at some of the newer ones. We're also going to talk about NMR and metabolite imaging after this, and then we'll discuss some of the discoveries and applications of NMR-based metabolomics. And then really for the rest of the series here, we're going to talk about analyzing data. And this is often the essential bottleneck, and it's one that I wanted to make sure people understand how that's done. So again, people who may have seen the earlier presentation, which was, I talked about some of the essential advantages of metabolomics. The fact that it's very stable instrumentation, it's non-destructive, it's very good for quantitative measurements. Sample preparation is really easy. It detects a lot of compounds that can't be readily seen by other techniques like GCMS or LCMS. You can work with living systems, and you can measure flux and reactions and kinetics. And then if you're trying to identify unknown compounds, NMR is the gold standard for doing that. But there are disadvantages for NMR-based metabolomics. So NMR instruments are expensive. It's not unusual to pay upwards of $1 million, and some of the largest systems are up to $20 million. The limit of detection for NMR is not great. It's about 5 micromolar. We've seen that in other diagrams I've talked about. The volumes are relatively large, as opposed to 10 microliters that you need for mass spec. And the fact that people have to do a lot of the manual work. Now, we've talked about some of the automation techniques, and I'll reiterate some of these here again. But the fact that these are significant disadvantages is leading to a trend, and that even though NMR-based metabolomics is growing exponentially, MS-based metabolomics is growing super exponentially. So if there's a war between MS and NMR, MS is winning. But in that regard, it's a friendly war, and I think, as I've been saying before, it's a matter of really using both techniques to their fullest. And really, this is more a reflection of the fact that MS technologies have advanced more quickly than NMR technologies. And it's really up to the NMR community and the NMR manufacturers to see if they can make NMR more competitive. And this is essentially a comparison between them, and I think we saw this before, but this is the differences between NMR and GCMS and LCMS. So the limits of detection are in micromolar, whereas in MS instruments it's a nanomolar. Number of compounds, 50, 60 for NMR, but it's up to several hundred with mass spec. In terms of manual analysis, most people are content if they can analyze about 20 samples a day by NMR. But with MS methods, it's not unusual to get up to 80 or 100 or even 200 samples a day. And because there's a lot of manual intervention with NMR, the costs per sample are relatively high. It's not a whole lot cheaper than the other technologies. So given those limitations, you'd say, well, why use NMR at all? So if you want to make NMR competitive, what's the call for NMR spectroscopists is to make the technique more sensitive, to increase the number of compounds, which comes with increased sensitivity, to increase the number of samples that can be processed, and to reduce the costs. So again, we've talked a little bit about how you can change that. And if you look at the general metabolomics workflow for NMR, there are tasks related to sample preparation, tasks for sample loading and acquisition, tasks for spectral processing, deconvolution and peak assignment, and data interpretation. And depending on whether you do 1D or 2D NMR, depending on whether you have complex samples, each of these can take anywhere from a few minutes to up to 10 hours. And so what you're trying to do, at least looking at the workflow, is to see if you can A, speed up the sample prep and sample loading, see if you can simplify or speed up spectral processing, see if you can improve or accelerate deconvolution and data interpretation. So increasing the sample collection has been done before, and it's already done by many manufacturers, whether it's sample jets or auto samplers. Certainly that accelerates that. We've talked about that. Spectral deconvolution. We've also talked about that, how it's critical and one of the bottlenecks in NMR spectroscopy. One that's typically done by manually approaches, manual software. So we've talked about AMICs and Kenomics that do this, and typically how long it takes with those, with even skilled people. And then I've mentioned the other automated methods that have just started coming out, developed by Brooker for wine and juice and honey, and also very recently for the analysis of lipid subclasses or lipoprotein. I've also talked about some of this automated deconvolution tools, from Batman to CTLS to Bayesil. And I introduced Bayesil yesterday, I guess, about what it does, how it works, and the principles behind it. So this is, again, an effort towards automated deconvolution. What I didn't mention, though, is that even with Bayesil being available, a lot of people don't follow the instructions. And so one of the things that we've ended up doing more recently is converting it more to a kit. So this is something that the molecular biologists understand. There's all kinds of protocols about how to do cloning and how to do PCR and how to do everything else. But if everyone tried to follow the written protocols, no one would get anything done. So what they do is they have kits. And so now in molecular biology, everything is very routine. So the same idea is actually happening already in the field of mass spectrometry and metabolomics. They have kits. There's starting to be trends for GCMS for kits, and so why not NMR? So what we've done is we've improved Bayesil a fair bit. We've made the software handle batch analysis. So instead of one spectrum at a time, as the web server allows, this is something you can upload lots of spectra. We've put it on a faster cloud computer system, so it's about 2 or 3 times faster. And then because computers aren't perfect, and occasionally you'll see things that you don't quite agree with, we've put an interactive editor, which is similar to what you'll see with Kenomics or AMICs. So you can adjust things if you don't like what you're seeing. And then there's more spectral libraries, more field strengths. But the most important thing that's in the kit is actually the reagents, which are needed for both phasing and referencing, as well as the ultracentrifuge tubes, which are critical for getting rid of the proteins, which apparently most people forget to get rid of. So with that, by using the kit approach, and by putting and bundling all the reagents and tools that you need to do it into one box, the sample cost is really down to about $10. So the kits are available through the Metabolomics Innovation Center. It has to make these things available at a non-profit rate because it's a service center. Eventually, these things will probably be sold through some other companies because it gets hard to keep it up. But the point is that it is getting cheap and fast to do NMR-based metabolomics. There are other ways of improving the speed at which you do NMR. So typically, if you're using a sample jet, some of them refrigerate the samples. But in many cases, people will put the sample as refrigerated into the NMR and wait for it to warm up for 10 minutes. You can also have a pre-warming stage in your sample jet system, and that will get the temperature to exactly the right temperature so that the sample can be shot straight in, and there's no waiting. So that helps improve the speed of sample acquisition. You can try and shorten your acquisition times and recycle delays. Obviously, the reference libraries have to be adjusted to that, but it is possible. Many people who traditionally work in proteins think about using perfect 90-degree pulses, but when you're working with small molecules and you're looking at high throughput, you don't have to do that. You actually can go for things like a 45-degree pulse or an Ernst angle, which allows you to pulse more rapidly, acquire more quickly. Zero filling is an important thing that's often done to improve your resolution, reduce some of the noise. Not all people do that, but it is wise to do that, and many of the software tools support that. There's also smarter apodization functions that you can use. So not just simple line broadening, but you can also use sine bell curves and other things that actually seem to enhance the signal somewhat. And then even with 1D, but certainly with 2D, you can start using things like maximum entropy or non-uniform sampling to reconstruct higher resolution, lower noise spectra. So there's lots of interest right now in the area of non-uniform sampling, especially in 2D and 3D spectra. This is something that's been going on, especially in the field of protein work, where you have only partial sampling along the 2D spectrum. But there are groups that are looking at just doing partial sampling along a 1D spectrum. And there's mathematical evidence that, in fact, that non-uniform sampling improves the noise, signal-to-noise rather, in the signal. So whether it's for 2D, 3D, or even 1D, it is possible to get a signal enhancement using non-uniform sampling. And in the case of 2D, it also helps with a reduction in time, up to a 4-fold reduction in time, and then a 3D on the order of perhaps a 100-fold reduction in overall time. There are other approaches that people are looking at. Again, it pertains to two-dimensional and even potentially three-dimensional NMR. And this is a technique called UltraFast NMR. And this is an example of, in this case, a COSY spectrum collected on a compound, fairly high concentration, probably millimolar. So it's a small molecule, and you can see the nice COSY spectrum. And then on the right is one that's been collected in 100 milliseconds. That's a 2D spectrum in 100 milliseconds. Now, it's obviously lower resolution, but that's a lot faster than you can collect a 1D spectrum. So what ultra-fast NMR does is use something or tricks that were originally developed for imaging. So the concept of echoplanar imaging, which was developed in the 1970s and for which a Nobel Prize was awarded, they simply applied that concept, the idea of using gradients and providing gradients along the spatial length of your NMR tube to encode the changes in the relaxation or signal changes for each component of the spectrum. So in essence, along the length of an NMR tube, you're measuring perhaps 100 different spin evolutions or little slices along the NMR tube. And so you're using those different evolutions to essentially act as the T2 component of the 2D spectral collection process. So that spatial information is then used in the 2D Fourier transform to produce the COSY, the HSQC, the TOXY, whatever. So it's an incredibly elegant approach, but it allows you to work with just a single sample and basically what seems like a single pulse. And that's how you're able to get 2D spectra in milliseconds. Now, if you want to go to N15 labeled, say, proteins, it takes on the order of maybe a minute. So the limitation with ultrafast NMR is that because you're doing this spatial encoding along the length of the sample, it means that you're looking at tiny voxels, if you want. So maybe a few hundred or a few tens of microliters. And so getting the spectrum for 10 microliters or 20 microliters is not going to be very sensitive. So the net effect is that it's relatively low sensitivity. There's also issues with limited resolution, limited spectral width. But with better technology now, things like cryoprobes, stronger pulse field gradients, you can overcome some of these problems, or in fact many of these problems. So that right now, really the only limitation is typically the diffusion of something that's homogeneous bouncing around that can cause some of the problems with the measurement. But this certainly tells you that 2D NMR can be as fast or even faster than 1D NMR. And if you could combine ultrafast NMR with non-uniform sampling techniques, then maybe the whole thing becomes down to tens of milliseconds to acquire things. So that's an example of a new technology that could certainly speed up metabolomics. Another thing to look at is increasing the sensitivity of systems. So if you want a more sensitive NMR instrument, you can pay more money. Bigger magnets give you better sensitivity. You can detect down to probably sub-micromolar if you're working on a 1.2 gigahertz magnet. But as I said, those cost about $20 million. You can spend less money and you can get a cryogenic probe, and that'll enhance your sensitivity by a factor of 4. So again, you can go from maybe 10 micromolar down to 2 micromolar. You can also improve sensitivity by working with microcoil probes or microprobes. And so this allows you to work with much smaller volumes, much smaller samples. And then in terms of a mass sensitivity, that also improves, perhaps by a factor of 5 or 10. You can always get more material, or you can concentrate things. So even though NMR people are loath to modify or work with their samples, it sometimes is smart to try and concentrate things. That sometimes costs a bit of money, but it depends on the sample you're working with. You can work with solvent-matched tubes, Shigemi tubes. Those are more expensive than regular tubes, but they allow you to work with much smaller volumes as well. A really, really cheap solution is to use tube-in-tube techniques. I'll talk about those a little later. So just as a picture, and again, some people may have seen this before, but there are ideal field strengths for certain types of fluids depending on their complexity. So juice is not particularly complex, maybe 30 to 40 compounds. Serum is a bit more, on the order of 50 to 60 compounds. Saliva, sometimes up to 60 and 70. Urine, usually around 150 to 200 compounds. And then maybe cell extracts from bacteria or plants might be 200 or 300. So as you climb up this ladder or staircase, you'll see that, in fact, higher fields are better for more complex samples. That spreads the signals out, but it also allows you to detect things down to lower concentrations overall. I'd mentioned the microcoils, the cryogenic probes, solvent-matched NMR tubes like Shigemi. All of those can improve your overall sensitivity because you're either working with smaller volumes or enhancing or reducing the noise from electronic noise and so on. Those cost money. Something that doesn't cost money is the idea of working with basically smaller tubes or smaller volumes. So you can work with 1.7 millimeter tubes, or you can work with 3 millimeter tubes, or you can work with 5 millimeter tubes. If you don't want to play around with getting a whole rack of new tubes, which costs a fair bit of money, you can actually put little capillaries inside your standard tubes. And then with these, you can actually get as little as 10, sometimes up to 50 microliters. And the nice thing about capillary tubes is that they're disposable, and that means that you won't break your other NMR tubes, which cost a fair bit of money. All you need to do is basically create a Teflon plug that's about 5 millimeters in diameter, and you can usually get a machine shop to make a whole bunch of these things, and they'll slide tightly and smoothly into your sample. And you can retrieve your capillary if you wish, and dispose the sample, because it's almost impossible to get something out of a capillary tube, but you can also store it for a long time as well. Now, it's not necessarily going to be high throughput, but if it's an issue of sensitivity and dollars, tube-in-tube NMR works really well. Another approach for enhancing sensitivity, and again, this is something that I brought up earlier in the presentations on Tuesday, was the idea of using dynamic nuclear polarization. So with this technique, you can enhance signals by 100 or even up to 1,000-fold, depending on a combination of temperature and radicals that you're using and the degree of microwave radiation. So the concept of DNP is to basically take a sample, cool it down a lot, irradiate it with microwaves, and essentially catalyze or enhance the transfer of electron polarization to nuclear polarization. So polarization with electrons is much better and much greater at low temperatures than it is with nuclei. And if you use things in the right way, it's possible to move those paramagnetic electrons and that polarization over to nuclei and get an enormous enhancement. So this is illustrated here with the relationship between the level of polarization. This is the population in the different energy states, N2 being the high energy state, N1 being the lower energy state. And the polarization is just the difference between the two divided by the sum of the two. And so there is a Boltzmann equilibrium that's dictated by temperature and energy differences. But if you keep the temperature down, then obviously you can increase the level of N2 over N1. But there's also a very characteristic feature about both electrons and protons and how easily they're polarized. So with electron polarization at 3 degrees Kelvin, you can get 95% level of polarization. At 3 degrees, proton polarization is about 0.1%. So a 1,000-fold difference at, say, 10 Tesla. So it's a function of both field strength, as I said, temperature, and the characteristic of in some cases, the electrons and the protons, sort of the chemical characteristics. But the idea of DNP is basically transfer all of that energy from the electrons to the nuclei, or the protons, and you get huge, up to 1,000-fold signal enhancements. So to play with electrons basically means you have to play with microwaves. And to play with NMR means playing with radio waves. So you can actually do that by connecting up a microwave system and an NMR system with radio frequency pulsing and put them into the same magnet and the same coil and the same system. So there are several types of liquid state NMR/DNP that have been developed. There's also solid state DNP, where you can put and keep everything in the solid state and use magic angle spinning. But again, you have to have both a microwave generator and an NMR compatible system. And then there are also methods that use what's called dissolution DNP, which is used for both MRI and NMR. And this is explained a bit more here. So typically you'll take a sample that you want to polarize. You'll cool it down to 1.2 Kelvin in this case. You'll put it in a big magnet, not a really big magnet, around 6 or 7 Tesla, which primes it up for the right conditioning, and then you will polarize with a microwave gyrotron. So this is happening obviously for something that's frozen solid. Not too many things are liquid at 1.2 Kelvin. What you then do is very quickly thaw the sample, turn it into a liquid. And that arrow essentially shows what happens. Is you take the liquid and you inject it straight into your regular NMR instrument, in this case 300 megahertz NMR. Now if you want to do it at 11 Tesla at 500 or 14 Tesla at 600 or whatever, you'll have to do have a stronger gyrotron and bigger magnets. And so it does become expensive. You can also, during the time that you're injecting samples, you can actually inject it into an animal because it's now liquid. And you can also monitor it during that rapid injection phase. So the standard protocol here is to take your sample, and it might be typically C13 or N15, so it's isotopically labeled and enriched. You put a special radical into it. So it's typically a tridal radical that helps facilitate that electron polarization because it's going to have a free electron to your carbon/proton nucleus of interest. You put it in what's called DNP juice, which is a mixture of glycerol and water. Cool it down so it's frozen, irradiate with microwaves, and let it sit there while it's being irradiated for about 3 hours. So it takes a while. Then you do a rapid temperature jump, so it melts. It's still highly hyperpolarized. And then you transfer it very quickly, in a few seconds, to the NMR or to the MRI or to the animal, which is in the MRI. Then you start acquiring. So it could go either into an NMR tube, a person, or an animal, and collect. And because it's so highly polarized, you don't have to collect for very long. So you've got this great signal. And this is just illustrating it here. So here's a carbon-13 urea labeled sample. It's in a mixture of glycerol water, that's the DNP juice, in the appropriate ratio. And you can see what happens basically if you just tried to collect C13 urea using conventional NMR. You can see the signal way down below, and that's been enhanced by a factor of 10 just so you can just see the signal. When you irradiate the sample and get this DNP transfer, the signal is enhanced by 60-fold. So that concept of enhancing and working with carbon-labeled material or N15 labeled material has actually been moved into metabolomics. And what they've done here is that they've taken acetic anhydride, which they cool down, put it in the DNP juice, irradiate with microwaves for 2 or 3 hours, and then rapidly dissolve it and rapidly react it with, in this case, a bunch of amino acids. So acetic anhydride will react within microseconds at almost 100% efficiency with free amines. So it doesn't have to be amino acids. It could be almost anything else that has a free amine. So it does an acetylation reaction. You now have a C13 label on your sample, and it's now hyperpolarized, or at least it still is. And so you'll get this roughly 60-fold enhancement of your carbon-13 signal. And because the acetyl group is so close to the side chains of the amino acids, it's sensitive to their side chain configuration, and so you get different resonances for each of the different amino acids. If this acetyl group was further away, say instead of the C1 but at the C2 position, you wouldn't see this. So it's really critical that the labeled carbon be very close to the side chains, at least 1 or 2 or 3 bonds away. But it's a remarkable level of enhancement, and so 60-fold. So if you take 5 micromolar as your lower limit, divide by 60, you're looking at tens of nanomolar, which is potentially possible with DNP. Now, the system that I've described that uses these hypercooling systems with multiple magnets, temperature jump systems, gyrotrons, they cost about $2 to $3 million. They're not cheap, and they require a lot of skill and a team of people. So it's not just something you can do at night on your own. But there is something you can do, or that's starting to happen, and this is this thing that I called dirt cheap DNP. And this has been developed over the last 2 years. And it goes by another name called the saber sheath approach. Now, if you look up saber sheath on Google, it'll actually probably point to a method for doing tracheotomies. But if you type in saber sheath and NMR, it'll talk about DNP. So the approach here is actually not to cool things down with a big cryogenic system down to 1.7 Kelvin, and it's not to put it in another magnet. In fact, it's to put it into a non-magnet or an anti-magnet, if you want. So what's done is you bubble a paramagnetic material, in this case paramagnetic hydrogen gas, which isn't too harmful, and you also put in an organometallic compound, typically an iridium type compound, which helps catalyze the transfer of the hydrogen paramagnetic ions to the recipient molecules of interest. So it's just a conduit, the organic metallic compound, and it can be soluble potentially in water or in mildly polar organic solvents. And so it takes the paramagnetic hydrogen signal, the electron signal, and moves it to the N15 or the C13 labeled material. Now, the interesting thing here is that you don't have to have that big magnet. In fact, you put it in a magnetic shield. It's called mu metal. It's a mixture of nickel and stainless steel. And the magnetic field in this shielded material is microtesla. So it's essentially a zero field. It's many times weaker than the Earth's magnetic field. In fact, the Earth's magnetic field is so strong it messes it up. So this is why you have to shield it. So instead of sitting in the polarizer system for 3 hours, you put it in this shielded thing for less than a minute. And then you take the sample out and transfer it into your NMR. So the entire process is done at room temperature, takes about 1.5 minutes, and costs less than a few $1,000s. The most expensive part actually is the shielded mu metal. So it's something you can do alone at night in the lab, but it's also something that gives you the same sensitivity enhancement as these $2 or $3 million instruments. So there's a tremendous amount of excitement about this in the field. There's a number of people that are working on it. Whether it's easily applicable to aqueous systems, still not clear, but there are many, many metabolites that in fact are nicely soluble in organic solvents. So it may be a mechanism or a route to start looking at a lot of other metabolites that are semi-polar metabolites and enhancing their signals by 100 or 1,000-fold with very little cost. So we've talked about improving the speed with the idea of automated deconvolution, kits if you want, more rapid 1D and 2D acquisitions. We talked about sensitivity enhancements using better magnets, better technology, or DNP. There's also ways of simplifying NMR spectra. If you can simplify NMR spectra, that also improves the speed and the accuracy in which you can do spectral deconvolution. So there have been a number of ideas floating around. As a rule, a 1D presat NOSY type experiment allows you to get the broadest coverage of most molecular types, both the proteins and the small molecules. And this has been known for a long time. And that's why the NOSY presat is almost the standard sequence used in every NMR 1D experiment. You can do T2 editing, and this is the CPMG, or Car per cell Meiboom Gill pulse sequence. This has been around forever, probably since the 50s, I guess. It allows you to get rid of the protein signals. And so you can compare on the left upper to the right upper, and you can see that the protein, the broad signals on the right-hand side are all gone. So it's impressive. It's not perfect, though, but it is impressive. You can go the opposite way, and you can do diffusion editing or DOSY type experiments, and that actually has the opposite of a CPMG. It enhances the lipid regions and eliminates or reduces the sharp peaks. And again, if that's what you're interested in, lipids or lipid proteins, this is a good choice. You can also do something that's quite elegant, which is a 2DJ resolved spectra. This is something that Mark Viant has been doing for a number of years. Hasn't really picked up much, but it is possible to get a 1D projection of a 2D resolved spectrum. And when you do that, it largely eliminates a lot of the couplings. So the complexity of the spectrum in terms of a 1D projection is greatly reduced. So this concept actually has been sort of extended over the last 5 or 10 years to an application called pure Shift NMR. And this is where you can take a complicated molecule which would generate lots of strong couplings and lots of bizarre coupling patterns and reduce it from about 30 different peaks to 3 different peaks. And these 3 different peaks correspond to the 3 different chemical shifts in this particular molecule. So, if an NMR spectrum has 1,000 peaks and you're able to broadband decouple everything, that 1,000 peaks can be reduced probably onto the order of 100, 150. It's trivial to deconvolute that. You can run, anyone almost could write a program that would figure that out. So what pure Shift NMR is a broadband decoupling proton experiment. You're decoupling across everything, which is almost impossible to do, but there's some tricks they've done. So what they do is they do frequency selective excitation during, again, a weak gradient. So you can do lots of things with gradients. So what you're doing is, in essence, you're kind of acquiring a 2D experiment, and I'll show you how, and reconstructing a broadband decoupled 1D experiment. And so it's complicated, and unless you know a lot about pulse sequence design, it won't make too much sense. But what's illustrated here is this sort of, you know, spin echo type experiment, but there's a gradient that's applied. And you increment the T1s stepwise, just like you would do in a 2D experiment. And then you acquire during the acquisition period over a very, very short time, just a few tens of milliseconds. And you repeat this. So what's illustrated there is where you're acquiring in the FID. So the first T1 increment, you acquire the data for the first 20 milliseconds. The second one, you wait 20 milliseconds, and you acquire from 20 to 40 milliseconds. The third one, you wait 40 milliseconds, and you acquire from 40 to 60, and so on. So then you can reconstruct the FID with these different T1 periods. And so you now have essentially an artificial FID, but it allows you essentially to have carried out this broadband decoupling. So there's a bunch of different tricks, different versions of the pure shift NMR, but they have basically the same concept. So the net result is you can get these great looking spectra, but just like with UltraFast NMR, it's relatively insensitive. Typically you have to have higher concentrations, and you have to acquire not just in a minute, but 10 minutes or 20 minutes, just like a 2D spectrum. But if you can get that level of spectral simplification, it's probably worth it. So, as we've pointed out before, 1D spectra, when all the couplings are allowed, are really, really complicated. And so you can simplify them through spectral editing, like the CPMG methods, or through techniques like the pure shift sequences. But you can also go to 2D. And 2D, whether it's a TOCSY or an HSQC, helps also to resolve non-obvious couplings, or in some cases reduce the couplings, in the case of HSQC. So with an HSQC, you don't see all these multiplets. You just see basically a single peak, which is a lot easier to interpret. So with 2D spectroscopy, you get much better resolution, so you get a gain. Even if you're working at lower field, you still get better resolution. You get information about connectivity, something we talked about, how you could do the same thing with a statistical TOCSY, but you do get atom connectivity, so you know which peaks correspond to which atoms or molecules. Generally get a more complete spectral fingerprint, so there's more information in a 2D spectrum. Allows you to collect not just proton shifts, but proton and carbon, proton and nitrogen, or if you go to higher dimensions, proton, carbon, and nitrogen. And then you can use the amplification of signals by having, at least if it's C13, you get enhanced signals for both carbon and proton if you do heteronuclear HSQC or HMQC experiments. So those are the advantages. The disadvantages are that they still, in most cases, in most people's hands, are time-consuming to collect. There's still a processing time or step, so it's not trivial. With 1D, it's a few seconds. With 2D, it's often a few minutes to sometimes an hour. There aren't a lot of spectral databases for 2D spectra. Quantitation is a challenge with 2D spectra. And when you're working with natural abundance carbon or natural abundance nitrogen, it's not very sensitive. So we've hinted at some of the ways of making or getting around this. We've talked about things like ultra-fast spectroscopy, which can accelerate the time. But there are also other elegant ones which are using tagging. So I mentioned the tagging approach in DNP, but you can use tagging without DNP. And this is something that Dan Raftery has been doing for a number of years. So if you have a C13 or N15 tag and you tag it close to sort of the unique site of a particular metabolite, you can actually get a unique chemical shift. And so tagging increases the isotopic abundance. You don't have to grow things in carbon, nitrogen media or spend thousands of dollars of growing things in carbon dioxide environments for plants or getting mice and rats adapted to eating C13 foods. The tagging also allows you to be selective. You can tag amines, you can tag carboxylates, you can tag alcohols, aldehydes, and ketones. And by selectively tagging these things, you're effectively reducing the complexity. So that helps with simplicity. And then in addition to reducing the spectral complexity by selection, you're also reducing the spectral complexity because you're down to basically one signal per metabolite, not 10 peaks or 20 peaks or 100 peaks. So this is illustrated with this approach that, as I say, Dan Raftery has done. You could use it with acetic anhydride, like we did with the DNP example. You can do it with N15 ethanolamine, which instead of tagging free amines, will actually tag free carboxylates. So roughly half of all metabolites either have free amines or free carboxylates. So between these two tags, you can get most things, or a good portion of metabolites tagged. And this is just an illustration of the type of spectra you can get with an HSQC N15 proton of serum and urine that have been tagged with ethanolamine. And then below that is the 1D spectrum of a urine. So with the human urine example, maybe there's 75 peaks. Most of them can be identified, recognized, and pinpointed. Whereas with the 1D NMR spectrum, you're dealing with, 2,000 or 3,000 peaks, which are pretty hard to identify. And what Dan Raftery has done is he's actually published a list giving a whole bunch of N15 proton chemical shifts for these ethanolamine tagged compounds. So he's done this for each purified compound, measured the spectrum, collected the HSQC, and so now you can just look it up. So this is trivial to deconvolute. It doesn't require the complicated hidden Markov models and other things that we talked about with Bayesil. He's also gone a little further, and he's also now created a tag like the ethanolamine, but he's now attached a choline group to it, so it's called cholamine. And what this does is target the free carboxylates, and it allows you to get an N15 tag to do NMR, but it also has a charged tag with the choline group that allows you to do mass spec. So he calls it a smart isotope tag, and it, again, is something you can do combination of NMR mass spec together. And this was published by his group in 2013. So in addition to doing smart things with pulse sequences and smart things with labeling and smart things with 2D spectroscopy, you can also improve your samples. So one of the simplest things that almost no one does is you can concentrate your samples. That'll improve your sensitivity, allows you to see more things with less time, and in the end saves a lot of costs downstream. But you can also do a variety of other kinds of sample manipulation, which might be, in this case, if we're looking at serum or plasma, trying to extract metabolites from this complex mixture of lipids, lipoproteins, proteins, and metabolites. So this is just illustrating the type of extraction results using different protocols. One is ultrafiltration, which gets rid of the lipoproteins very nicely. Acetone extraction, which gets rid of lipoproteins pretty well, but also a few other metabolites. Acetonitrile, which historically used to be the one commonly used, but also doesn't quite get rid of lipoproteins. Perchloric acid, which damages your sample quite severely. And then methanol chloroform, which selects primarily hydrophobic metabolites. So a paper was published in 2008 looking at these protocols and comparing them. And the answer was pretty clear from this study, and that was ultrafiltration is your best method. And that's the method we've been using ever since, and many other groups use. And that's the method that's configured for Bayesil and the Bayesil kit that I talked about. A close second is sort of the acetone extraction, but again, there are issues with that, and it drags around as a contaminant that you'll see in your NMR spectra. More recently, Dan Raftery has revisited this, so this was just last year, and rather than working with acetone or acetonitrile, he said, well, why don't we just try methanol? So it's the one solvent that is close to water, or as close to water as you can get, pretty much. And they were able to avoid a lot of the problems with both the toxicity of acetonitrile, but also some of the issues with ultrafiltration. What it does, actually, is it releases the metabolites from serum albumin. And it leads to precipitation of the proteins. And by releasing a whole bunch of metabolites from serum albumin, you actually increase the number of compounds that you typically see. So with ultrafiltration, most of the time you get around 50 to 55. With this approach, you get up to 70 compounds that are released. What's more, with the methanol precipitation, it's much more suited to high throughput work. You can take 100 samples, put in so many drops of methanol in every sample, spin them down, and then there is a freeze-drying step, but you can freeze-dry 100 samples simultaneously pretty easily. It's hard to spin down 100 samples with a single microfuge. So a number of groups are starting to adopt this. We're a little hesitant about adopting it, partly because the normal values for metabolites that are quoted in the literature are for free metabolites, not the ones that have been released by serum albumin. So if you're using reference values from clinical papers or journals or clinical labs, you'll get different values with this approach. On the other hand, if everyone adopts this or everyone is clear about it and says this is methanol extracted serum or methanol extracted plasma, that's fine as long as it's prefaced with that. And then if there are values that are measured or healthy and abnormal and we get those values, then this could be perfectly fine. So as I say, we're sitting on the fence, but it's an exciting new development, and I think certainly improves the number of compounds that you can get, and also the limit of quantification is improved as well. In addition to playing around with methanol and simplifying things, or concentrating, just using standard filters or SPE columns, you can also simplify NMR by using silica nanoparticles. So we're in a nanotechnology facility here, and I'm sure they have these all over the place. Anyways, nanoparticles have a large surface to volume ratio. They can be charged or they can be neutral. In the case of silica nanoparticles, they have a very strong negative charge. They're easily dispersed in water, easily added, safe to use, probably not safe to eat, but they can be easily added to an NMR tube and you won't see any ill effects. When they added this, and this is Raphael Bruchweiler's group, and I think it was accidental, but anyways, they dropped it in and they noticed something striking, which was when they added the silicon nanoparticles, the spectra got much simpler compared to the one on the left, which has lots of peaks, and the one on the right has fewer peaks. And it wasn't just a case that there was some precipitation going on. And in fact, what was happening was the silicon nanoparticles were binding positively charged compounds. And they weren't touching the negatively charged compounds. And this is because silica is strongly negatively charged. So what's happening is that you have positive to negative attraction, and those positively charged compounds are now stuck to this giant nanoparticle, which is rotating slowly. And so this will give you essentially very, very short T2s, or very, very wide peaks, so wide that they essentially disappear. So all that you can see are the peaks that have long T2s that give you sharp peaks. So this, again, is just sort of blowing things up, and you can see with the SNPs, fewer peaks, without the SNPs, quite a few peaks, and the things that essentially were removed, the compounds that were positive, so arginine, lysine, histidine, were pulled off, whereas citric acid, lactic acid, shikimic acid were stuck around. Now, it's not perfect, and playing around with the pH and other solvent conditions allows you to see some more than others. But in essence, this is what's going on here, which is this differential relaxation, or T2, things that have fast tumbling rates or long T2s are NMR visible, and the ones that have short T2s are essentially NMR invisible. So that's an example of sample manipulation. You can play around, as I say, with other types of nanoparticles, ones that are positively charged, ones that have gold surfaces, ones that may have other kinds of reagents attached to them, which may have some affinity for certain metabolites. And again, this allows you to selectively remove certain metabolites just by progressively adding different types of nanoparticles. And it really doesn't perturb the system. So I guess to summarize some of the key developments with respect to new techniques. So the first one we've highlighted are the faster automated spectral deconvolution techniques, whether it's Rooker's development, semi-automation with Conomics, full automation with Bayesil. The idea of kits, which I think we're going to see more and more of. It's already happening in mass spec, starting to happen with GCMS, starting to happen with NMR. I think we're seeing obviously better autosamplers, smarter autosamplers that have temperature control, which is very important for especially metabolomics and for more rapid acquisition with the thermal warming or pre-warming. Smarter 1D acquisition and processing. There are always little things you can do to make the acquisition and processing a lot faster. We've talked about non-uniform sampling methods, which reduce the amount of time to collect 2D and 3D spectra, and potentially even improve the signal to noise for 1D, 2D, and 3D spectra. We talked about ultra-fast spectroscopy, which uses this spatial encoding technique, a very elegant approach where you can get spectra in a matter of milliseconds and potentially heteronuclear spectra in a matter of a minute or less, so comparable to the best you can do in 1D spectra. There are always improvements to magnets and probe technologies, micro and nano probes. Those are expensive, but in many cases worth it. If you don't have the money or waiting for a new magnet, you can do things like tube-in-tube NMR, which costs pennies. We talked about dynamic nuclear polarization as an approach, which is very hot right now if you have the money. If you don't have the money, saber sheath is emerging and something that I think people should give a try. Things that have been around for a long time, smart spectral editing has certainly been around. CPMG kind of works, but I think there are better methods, things like pure Shift NMR, better routes for deconvolution. I think playing with your samples, doing C13 and N15 selective isotopic labeling, as Dan Raftery has done, certainly opens the doors to actually seeing more compounds, and then applying a lot of the techniques like DNP, like 2D NMR and UltraFast NMR to your samples. And then there's other improvements with sample manipulation, whether it's concentration, ultrafiltration, SPE columns for extracting things, or using silicon nanoparticles to simplify your spectra. All of those help. So in terms of where are we now, certainly with the automated deconvolution techniques, we're up to 150 samples a day and costs around $10. With improvements that Dan Raftery has described, along with some of the other ones we've mentioned, certainly we're trending down to below 2 micromolars. If you have really big magnets like you do here, perhaps down to micromolar, submicromolar. And then if people could get DNP working, or some of the other techniques that I've described, we could actually be in that realm of getting close to 100 nanomolar sensitivity. So if all the stars align, or if continued advancements happen in both NMR and in the lab, I can see the day not too distant from now where, in fact, NMR meets all these goals of having that detection limits of maybe 100 nanomolar, where it's routine to see and measure and quantify 150 to 200 compounds. And maybe the throughput will continue to climb to above 150 samples a day and continue to drop in cost. And at this stage, even now, I would argue that NMR is now quite competitive with any other technology. And It shouldn't be done to the exclusion of the other technologies, but certainly it should be of prime consideration when anyone's doing metabolomics, and should be part of, I think, any metabolomic processing plan. So with that, we'll wrap up.